Kannan sir?
Yeah, ma'am. Is this audible?
Yes, sir. Audible, sir. Sir,
Ma'am, can you give
Bhaskar?
Dr. Bhaskar as a co-host?
Yes, sir.
Yeah, ma'am.
Pratibha? Bhaskaran, sir. Pratibha, ma. Are you ready, ma?
Yes, ma'am.
Yeah. Okay, we shall start with session 2.
Shall I proceed, ma'am?
Yes, ma'am.
A pleasant afternoon to one and all present here. The world is full of diamonds and gems, and we are having some of them today to build this international conference on sustainable development goals in biosciences organized by Vikas and Vias. I would like to give my heartiest welcome to you all for this grand occasion. Let's start the technical session 2. Beauty is the good letter of introduction. Here, I invite Dr. M. Kannan sir, Head Department of Botany, Vikas, to give the guest introduction.
Yeah, good. Thank you, ma. Is this audible?
Yes, sir.
Yeah, good. Good afternoon to everyone. I am profusely overjoyed to take the opportunity to introduce our keynote speaker for this session. His name is Dr. X. Bhaskaran, a research scientist, Sun Yat-sen University, China. He is very humble and a good person. He has a great knowledge in the field of ferns. He completed his MSc Botany degree in Bharathidasan University in the year 2007. He completed his MPhil Botany degree in Periyar University in the year of 2008. He completed his PhD Botany in Bharathidasan University in the year of 2014. He completed his postdoctoral fellow in the year of 2017 at Sun Yat-sen University, China. Now he acted as a research scientist in same university in the year of 2015 to still till date. The following projects were completed by him, mainly Institute of Forest Genetics and Tree Breeding, Coimbatore, Ministry of Environment and Forest, Government of India. He acted as a project assistant in the year of 2007 to 2008. He acted as a junior. Yes, ma'am.
Kannan sir, could you on the video?
No, ma'am. It's some problem.
Okay, sir.
Okay, ma'am.
Thank
He acted as a junior research fellow in the year of 2008 in Salim Ali Center for Ornithology and Natural History, Coimbatore. He acted as a research associate in the year of 2014 to 2015 in Fairy Lake Botanical Garden and Chinese Academy of Sciences, China. And he have a vast knowledge, research scientist, principal investigator in the project of transcriptome sequencing and histochemical localization of vivipary birds of early tracheophyte fights in the year of 2015 to 2017. He acted as a postdoctoral fellow in the year of 2017 to till date in Sun Yat-sen University, China. He has rich knowledge in the following research field: transcriptomic, proteomic, and metabolomics, conservation, and pharmacology. He completed 6 book chapters. He delivered 8 guest lectures. He submitted 5 manuscripts and 8 manuscripts under preparation. He published his 26 research articles in various journals in national and international level. He also acted as a reviewer in 10 journals. He deposited 8 nucleotide sequence with NCBI. He presented his 16 research findings in international conference and symposium. He participated 17 conference, seminar, and also participated 20 workshops in national and international level. He got more than 5 awards, grants from various research institute, both national and international level, mainly DST Young Scientist Travel Grant in the year of 2010 November, DST New Delhi to China, and Outstanding Student Award by International Botanical Congress, IBC, 2017. On the behalf of Vivekananda Educational Institution and Vivekananda College of Arts and Science for Women Autonomous, Vivekananda Arts and Science College for Women, Sangagiri campus, cordially invite you the second international conference on sustainable development goals in bioscience. Welcome you, Dr. X. Bhaskar. Thank you.
Thank you, sir. The science of today is the technology of tomorrow. Now, I hand over the session to our chief guest.
Bhaskaran sir.
And now only audible. I think he have some technical issues. Yeah.
Bhaskaran sir, do you have any difficulties to share the screen or unmute the audio and the video?
Sir, any support, sir? Kannan, sir?
Yeah, ma'am.
Any help, sir?
He needs some help. He has some technical issues. Wi-Fi la ஏதோ problem அப்படின்னு சொல்லி சொன்னாங்க.
Okay, sir.
We can wait 5 minutes, ma'am.
Yeah, definitely.
Yeah, ma'am.
Kannan. Yeah,
Dr. Bhaskaran,
can you
sir?
hear me?
Now only we hear.
Yeah, okay.
Yeah, start now.
Yeah, okay. Thank you. So, First, I would like to thank the organizing committee of Second International Conference on Sustainable Development Goals in Bioscience, organized by the Department of Bioscience of Vivekananda College of Arts and Science for Women. And I also thank Dr. Kannan for a nice introduction about me. And today my topic is apomictic pteridophyte and its mechanism. As we know well, there are different types of plants in the plant kingdom. First, there are two different types of plant groups. One is non-vascular plants and the second one is the vascular plants. So in the non-vascular plants, algae, which includes algae, fungi, and bryophyte, doesn't have any xylem and phloem. And in the vascular plants, there are they also have xylem and phloem, which is present in the pteridophyte, gymnosperms, and angiosperms. So as we know, well, the ferns appear in the fossil record 360 million years ago, and it's much more important to study the evolutionary relationship of plants, and especially the evolution of woods, seeds, pollen, flowers, and fruits, because it's present in between the vascular plants and also the non-vascular plants. So that's why it's much more important to study the evolutionary things in the science. So the life cycle of pteridophytes, we know that ferns are producing the spores. It's released from the sporangium and it grow into a small and a hard type of young gametophyte. So in the young gametophytes, it's also producing the male antheridium and archiconium. So the antheridium is producing the sperm cell and the archiconium is producing the egg cells. So after the fertilization process, it forms the zygote. And after the zygote, it's produced the small sporophyte by the process of mitosis and mitosis cell division. And it's grow into in a mature stage, and it's producing the sporangium. And the sporangium, within the sporangium, it's producing several number of spores by the meiosis cell division. And from the sporangium, the spores are released. It's releasing the spores, and it will reproduce themselves. It's a normal life cycle pattern of pteridophytes. But now we are going to talk about the apomixis. The apomixis is asexual reproduction. So what is meant by asexual reproduction? Mean without any fertilization. So what is the fertilization? As I told you earlier, the fusion of antheridium and archiconium, the egg and the sperm cell fusion. This process will not happen in the apomixis. That is producing the seed without the benefit of the union of the sperm and egg cells. That is a apomixis process. And there are in all the plant kingdom group, especially in the gymnosperms and the angiosperms and bryophytes also have the same type of characteristics. In the gymnosperms, Biota orientalis, Cupressus torulosa, and Ginkgo biloba, Podocarpus makoi, Podocarpus macrophyllus, Epitra, Trifurca, Pinus valiciana. In the angiosperms, mangroves, Calonjo Dichrymontiana, lives in Coracana, Setaria viridis, and Monocat and Poaceae family. Among in the bryophyte groups, the gemma cup is formed, and also the adventive branches of an apomixis are also reported in the Marcantia. So these are the photos. The figures displays the apomictic characters of the plant kingdom. First one is the gymnosperms, and the second one is the fern. Uh, sorry, the angiosperm, and the third one is the gymnosperm, fern, and the bryophytes. So the vivipary in ferns. So in vivipary process, there are two different types of vivipary. One is a true vivipary, and another one is the pseudo vivipary. But in the true vivipary, megaspore remains within the sporangium, germinates. So within the sporangium, the spores are germinate and it develops into a gametophytes. And fertilized, it's reported in selaginal species. That's a true vivipary, like a mangrove species. And in some fern genera and families produce viviparous plantlets by asexual reproduction, that is a pseudo-vivipary on sporophyte fronds of Asplenium, Adianthum, Camptosaurus, Diplodocus, Tectoria, Woodwardia, Cyathea arborea, Dunstedtia, Grammaticeraceae, Hymenophilaceae, Viteriaceae. So there are so many ferns are having the viviparous characters from different types of family Atriaceae, Pteridaceae, Aspleniaceae, Dryopteridaceae, Thelipteridaceae, Pteridaceae, Dryopteridaceae, Tecteriaceae, Leptinaceae. I have found out these viviparous ferns in China during my postdoctoral research, and these are the photos showing the pseudo-vivipary ferns, pseudo-vivipary characters in different types of genus, Atrium, Asplenium, and Bolbitis, and Ampelopteris, Hemionitis, and Polysticum, Tecteria, Uvadia, Diplasium, Adianthum. These genus showed the viviparous characters in our greenhouse. I collected from the various mountains of China, and so first thing is I studied the spore germination of this Woodwardia. I selected two different types of Woodwardia species. One is the asexual reproduction character. It's having the Woodwardia orientalis is the asexual reproduction character, and Woodwardia japonica is having the sexual. It's a normal sexual character. So it's reproduce themselves by the sexual reproduction. The Woodwardia orientalis is reproduced themselves by the asexual reproduction. I compared these two fern species, and fertile fronds shade dried for 2 days, and the spores were collected. Collected spores preserved at 4 degrees Celsius for our analysis. And the spore germination, I used the MS medium, and also along with different plant growth regulators like ADA, DAP kinetin, GA3, and TD set. And spore germination was scored after 6 days for Woodwardia orientalis. It's a vivipary, the asexual plant, and 21 days taken for Woodwardia japonica of their inoculation. So spore germination after 6 days and the gametophyte growth rates were assessed after 60 days. So in the spore germination, after 6 days, the Woodwardia orientalis was 24, just showed only 24 percentage, and the Woodwardia japonica showed 95 percentage. So the sexual reproduction, the normal sexual reproduction ferns showed highest spore germination capability, and the asexual plants showed only lowest percentage of spore germination. So these are the photos explain the spore germination of a non-viviparous fern. I mean the sexual fern, the Woodwardia japonica in various hormones. And the second slide, spore germination of a viviparous fern. That's a apomictic fern, Woodwardia orientalis in various hormones. And these are the spore-derived gametophyte size of Woodwardia orientalis and Woodwardia japonica after 60 days. The length and width of the gametophytes were analyzed. So these are the photo showed the gametophyte development of sexual and asexual ferns in various type of plant growth hormones. And the transcriptome sequencing. So two Chinese medicinal ferns, namely Woodwardia orientalis and japonica, were grown in our garden. And a small amount of pinna is typically collected and frozen in liquid nitrogen and stored in minus 80 degrees Celsius for both RNA sequencing and qRT-PCR. Total RNA extracted using the OmniPlant RNA kit. So these are the flow chart of experiments. We used the Illumina sequencing, and we also further processed our raw reads, and alignments were also done. And we have raised the geo enrichment analysis and pathway enrichment analysis, expression pattern analysis were also done. So sequencing output of two various species. So in the apomictic fern, Woodwardia orientalis showed the highest number of reads, and also the Q20 percentages, and also the GC genomic content also measured. So among two different types of species, the asexual plants showed so many numbers of total rates due to several mechanisms it's going on within the asexual for their asexual mechanisms. And the gene sequencing of Woodwardia orientalis leaves and the vivipary buds. So here we have tabulated the read numbers, and the percentage were also given in the table for our and the SSRs were also analyzed. A simple sequence report were also analyzed. So the Woodwardia orientalis showed the highest number of SSR, the transcriptome, the transcripts were observed in the Woodwardia orientalis. The sexual plant showed only lowest number of the transcripts, I mean the SSRs. So, and the fourth table is statistics of SSR identified in the two Woodwardia transcripts. It's a di and the tri and the tetra and the penta and the hexa SSRs were reported here. So the distribution of the simple sequence repeats and the single nucleotide polymorphisms in two different Woodwardia species were tabulated here. So we compared the the asexual plant, I mean the vivipary fern, Woodwardia orientalis with the whitus vinifera, Theabromococca, and the Oryza sativa, Arabidapicis thaliana, Cucumis sativus, Solanum lycoborsicum, Clycin max, Fragaria vesca, and the Cissus originum, and the last one is with the Salaginella mollentorti. The highest the protein database was showed with Vitis vinifera in with Woodwardia orientalis, and in the Woodwardia japonica, when we compare with Theabromococca, it showed the highest number of protein database with Theabromococca. And second one is Vitis vinifera, third one is the Oryza sativa. So we compared with other group of plants. So it's a COG, cluster of orthologous groups. So it's a functional categories, and how much we observed 20,678 unigenes were observed to play a functional things in the Woodwardia orientalis, and only 14,212 unigenes were observed for Woodwardia japonica. So the flow chart shows the SOG of the vivipary fern. So it's a annotation of viviparous fern and non-viviparous fern. It's Woodwardia orientalis and Woodwardia japonica. So the total unigenes, so the highest unigenes were observed in the Woodwardia orientalis, and the lowest unigenes were because several genes are performing different types of function in the mechanism of asexual reproduction. So that's why the vivipary fern showed the highest number of unigenes. So here the distribution of E value in COG and cis brot and KIC KEGG pathways. So we observed some important genes in vivipary fern. The first one is the transcription factor viviparous 1, and the second one is the NOT3, NOT2, NOT3, NOT5 family isoform 2. It's derived from it's also present in the Theobromococca, and the abscisic acid deficient 4 unigene also observed, and dehydrine genes, leucoanthocyanidin dioxinase like genes, Della protein dwarf 8 like genes, and farnesyl pyrophosphate synthase 1 like genes, 9-cis epoxy carotenoid dioxinase, serine 309 protein kinase, abscisic acid receptor PYL4 like gene, gibberellic induced protein 1 deoxy D-cellulose 5 phosphate synthase, zeaxanthin epoxidase 1 deoxy D-cellulose 5 phosphate, 1 deoxy D-cellulose 5 phosphate synthase genes were also observed. So the RPKM value, it's reads per kilo base of exon per million mapped reads. So here the highest, we compared two different types of ferns, I mean the vivipary fern and the non-vivipary ferns. RPKM values were compared. So the highest percentage was observed, most of the genes were observed in the vivipary ferns, and some genes were present highly in the non-vivipary ferns, like I mean here we have compared two different types of things. One is the matured leaves and another one is the vivipary buds. So in the matured leaves, is present in the vivipary fern, and the vivipary buds it's present on the matured leaves of Woodwardia orientalis. So here, so what are the function is going on, and which gene is responsible for a particular function? So here the NOT2, NOT3, NOT5 family isoform 2 genes is responsible for the vivipary mutant genes. So this gene is inducing the vivipary character in Woodwardia orientalis, and the abscisic acid deficient 4 gene is reducing the ABA content, and the transcription factor viviparous 1 gene is responsible for the spore dormancy reducing gene. So here the spore dormancy was reduced in Woodwardia orientalis, that is a vivipary gene, and in the sexual reproduction and in the non-viviparous uh, fern, Woodwardia japonica showed 21 days of spore dormancy. And the dehydrin protein is performing the cellular protection against osmotic stress. And Della protein dwarf 8 reduces and represses gibberellic acid inducible genes. It's present in the viviparous fern. But all those genes are not present in the non-vivipary ferns. It's only present in the vivipary fern. I already told you that here the WOL. The WOL is Woodwardia orientalis matured leaf, and the WOV is Woodwardia orientalis vivipary buds. So the small buds are present on the matured leaves of Woodwardia orientalis. But these genes were not identified in the Woodwardia japonica, that is a non-viviparous. So these genes are responsible for the asexual reproduction in the Woodwardia species. UNC93-like gene resided in female reproductive structure and Apiconia as well as egg cells. The various expression patterns of UNC93, such as a temporal and spatial, implicate the apogamy or asexual induction in Ceratopteris richardii. So the UNC93 gene is also present in the Ceratopteris, that is another asexual plant. It's already reported in the earlier report. So we compared with the Ceratopteris richardii, and several genes may implicate in zygotic embryo development, and while it's mutate which induce asexual embryo development by various authors. So the presence of UNC93 like protein genes in Woodwardia japonica and Woodwardia orientalis. Uh, First one is the Woodwardia japonica, and the second one is the Woodwardia orientalis matured leaf, and we compared with the Woodwardia orientalis vivipary buds, and again we compared the matured WOL, I mean the matured leaf of Woodwardia orientalis with the Woodwardia orientalis vivipary buds, and these genes and most of the genes were downregulated, and these genes were upregulated. So this is the histogram of upregulated and downregulated iono genes between matured leaves and vivipary buds of Woodwardia orientalis, the apometric one. So most of the genes were upregulated. Most of the genes were upregulated, and only 4,989 genes were downregulated. So it's a duo of DEGs between differentiate expression genes between matured leaf and tributary buds. So it's a gene ontology classification. So in the histogram, the red lines, the red boxes shows the up regulation, and the green shows the down regulation. It shows the various function, metabolic process, cellular process, and different biological process, cellular components, and molecular functions. So different various types of genes are performing all these three functions in the Woodward ovarian of vivipary buds. So it's a here is the seed storage proteins and several globulins such as legumins, vicillins, globulins, P54 proteins are the primary seed storage proteins in dicoplants, and all of them destroyed on a germination, which applies to the seedlings as a early food material. Because when it's germinate, the embryo is replacing those replace these storage proteins, and two transcripts encoding glutenin is uniformly down-regulated, and the vicillin is also down-regulated. So as I told you earlier, the seed dormancy level is very low in the vivipary funds. So that's why it's selecting the asexual method of reproduction. So here the seed storage protein level is also down-regulated, and totally 20 transcripts are upregulated, and showing very low to very high expressions, while 18 transcripts are down-regulated, showing very low to moderate expressions. So we compared the expression level based on the RPKM value. So between 22 genes of 16 proteinase, 21 genes were upregulated and one of them was downregulated. 16 the proteinases was involved in the senescence, programmed cell death, seed storage protein progress, and their movements in signaling pathways, as well as for the biotic and abiotic stresses also it's involved. And the spore abortion in Woodwardia orientalis. Abortion of seed happened at the various bases of embryonic growth, programmed cell death close to both plants vegetative and reproduction growth. As both cysteine and aspartic proteinases are required proteolytic enzymes for PCD, the programmed cell death. So many genes are present in the Woodwardia orientalis for the spore abortion. Because when I observe under the light microscope of spores of vivipary ferns and the non-vivipary fern, the Woodwardia orientalis and Woodwardia japonica, so many spores were aborted in the Woodwardia orientalis, because these genes are present in the Woodwardia orientalis, and only very few spores were aborted in the. It's a normal, but so many numbers. Nearly 90 to 95% of spores were aborted in orientalis, but only very less percentage of spore were aborted in Woodwardia orientalis, Woodwardia japonica. So here are the spore abortion genes in Woodwardia orientalis. leaf and Udaria orientalis vivipary buds. So aesthetic proteinase were regulated. Aesthetic proteinase A1 like genes. These genes were down-regulated, and WRK transcription factor genes were also upregulated. And here cysteine proteinase genes mostly upregulated in the Woodwardia orientalis. So the seed development and what are the genes are present in the development of seed? Cumin family storage proteins. Uh, so we find out the globulin proteins, oleosins, late embryogenesis, abundant proteins, globulin proteins. It's a key storage proteins in the matured embryo. In Woodwardia orientalis, there are two different types of globulins, 7S and 11S. So moreover, most of them are down-regulated, which shows significantly increased expression. So the seed development genes were down-regulated in the vivipary form. So that's why it's selecting the asexual method. And also the seed dormancy level is also lower. That's why. And on the other hand, two genes were upregulated and showed decreased expression. So here the genes, I mean the legumin and immunoglobulin and basic 7S globulin genes were upregulated, and the legumin, the two genes were downregulated, and the late embryogenesis abundant genes were downregulated. So see here, most of the genes were downregulated than the upregulated genes. So here also the oleosin gene and the seed maturation proteins and the alpha amylase, beta amylase, transcription factor MYC2 gene, and the target of rhabdomycin isoform 1 gene. The rhabdomycin is also helpful in the seed development. So most of the genes are down-regulated, so that's why it's selecting the vivipary character. And the calcium may be an important second messenger in abortion regulation. So in general, calcium might play an vital role in a second messenger in abortion regulation. So in our study, several genes were upregulated. It's calreticulin, calmodulin, calcium-dependent protein kinase, phosphodiesterase, calcium-binding proteins, CDL, interacting serine threonine protein kinase genes, and calcium transporting ATPase genes were upregulated with different expressions of RPKM, and only few genes were downregulated. Calmodulin, calcium-dependent lipid-binding protein, and protein kinase, calcium-binding protein, calcium transporting ATPase were down-regulated. And the genes involved in the reactive oxygen species. So several genes like respiratory burst oxidase, reticulin oxidase, catalase peroxidase and peroxidase, catalase superoxide dismutase, ascorbate peroxide, ethylene response factors, glutaredoxin, glucanases, glutathione transferase, polyphenol oxidase were upregulated, and the respiratory bus oxidase, peroxidase, catalase, ethylene transport factor, glutaredoxin, glucanase, phenylalanine ammonialase genes were downregulated in the liver. So here the plant hormone biosynthesis. So in our study of transcriptome sequencing, the lowest expression of aldehyde oxidases were found in vivipary fauna. The aldehyde oxidase gene is the final stage of gene to produce the abscisic acid. So according to Farnsworth and Foran, uh, the abscisic acid levels lower in mangrove vivipary plants uh, than the embryos of non-mangrove and non-viviparous sister taxa. So the biosynthesis of ABA content is start with the zeaxanthin, and it's produced as a all trans violaxanthin. It's divided into two groups, and all trans neoxanthin, and here another step is a 9-cis violaxanthin. So this one, both of them are producing the xanthosine, and from the xanthosine, it goes to ABA aldehyde. The ABA aldehyde gene is producing the abscisic acid hormone. So in our observation, as I told you earlier, the lowest expression of aldehyde oxidase was found in the vivipary one than the non-vivipary one. I also quantified the ABA hormones. The hormone content level was also lower in the vivipary one than the non-vivipary one. So here, the Woodwardia japonica is having different types of aldehyde oxidase gene, and you can also see you can compare the KM level of Woodwardia japonica and Woodwardia orientalis. See here the expression level is higher. Here only one gene shows the highest one, but I mean the two, but the others are the lowest. But here it's also lower. The expression level is higher. It's 16.44. Here it's only 13. Why plants form vivipary character? Is any special substances? Yes, it's plant hormones. The German botanist Julius von Sachs. The plants produce, transport, and perceive organ-forming substances for the formation, growth, and different plant organs. So the various plant hormones plays a vital role in the plant growth. The abscisic acid is a universal hormone. It's present in all the group of plant kingdom. It's mainly to the positive regulation of dormancy, induction, and maintenance. And the second one is the gibberellic acid. It releases the dormancy, and it's promote the germination and counteracts the ABA effects. And the third one is the ethylene, promote the germination, counteracts the ABA effects. Brassinosteroids, it's promote the germination and also the counteracts the ABA effects. So what is the cause for vivipary? That's the loss of spore dormancy and the lowest level of ABA content. So by the use of seed germination or the spore germination of viviparous ferns and non-viviparous fern, as I told you earlier, the lowest content of abscisic acid were observed, and the spore dormancy was also lost because the spore germinated within 6 days in viviparous fern, and the spores were germinated in 21 days in non-vivipary fern. So the role of ABA in vivipary. So the abscisic acid has great importance in the development process, induction of seed dormancy, protein lipid synthesis, tolerance to desiccation, and inhibition of embryo to vegetative development. deficient in ABA production or sensitivity fail to enter presence resulting precocious germination. The precocious germination is an early germination. As the novel, the mangrove seeds can germinated early when it's attached with the mother plants. The mangrove seeds can germinate it. That's a precocious germination, or in other words, it's also called as viviparity. So the dry conditions increase the abscisic acid levels. The condition is not favorable for the viviparity. It's also found out by so many authors. So here I have tabulated the ABA biosynthesis and viviparity related genes in plants. So for the ABA metabolism, so many genes are performing the function, and the ABA transport, ABA signaling, calcium dependent factor. So different genes performing various levels of mechanism for the metabolism, transport, signaling of the viviparity forms. And in the viviparity mutant class, so here the viviparity mutant deficiency and insensitivity. Even these genes, if these genes are present, sometimes it may be a mutant gene, sometimes it may be a deficient gene, sometimes it may be an insensitivity gene. But whatever it may be, the seed effect shows the viviparity characters. The seed dormancy level is reduced due to the presence of these genes in the plant groups. An impact of viviparity and ABA. The viviparity and inhibition of pigment expression are caused by a pleiotropic action of a single gene. So the pleiotropic action means so many functions can be controlled by a single gene. That is a pleiotropic action. And a single metabolic block might induce both the carotene deficiency and also the viviparity. So these genes are also deficient to the carotene, and also it's also reduce the anthocyanins, and it also reduces the content of various metabolites like cinnamic acid, caffeic acid, and coumarin. So impacts on secondary metabolites and the protein. So the ABA genes are also so many. I mean the anthocyanin 2 dihydroflavanol 4 reductase and leucoanthocyanidin dioxygenase genes are involved in the flavonoid biosynthesis. It's also responsible to the viviparity-1 and ABA biosynthesis. So the seeds of ABA deficient and ABA insensitive mutants of CMAS and Arabidopsis exhibit desiccation intolerance, reduced protein accumulation and lack of dormancy, and it leads to the viviparous germination. So all those things we literally we collected and we compared with other group of plants. And inhibiting ABA signaling, ABA shows the strict requirement for ABA signaling and modified by the presence of VP1. So the VP1 gene is performing as a pleiotropic gene. It's a control all the functions of the AP, I mean the viviparous character of the plant. And 60% of the ABA dependent class was ABA inducible, but the induction was inhibited by VP1. Due to low ABA content and viviparity inducing genes, plants produce viviparity. I mean, it's asexual reproduction. So here, uh, the genes are responsible to the dehydration we observed 3 genes were upregulated and 7 genes are downregulated. So, as I told you earlier, some genes are also, due to the presence of the vivipary genes, the metabolite content is also affected. So the flavonoids have several functions such as abiotic stress production, for the UV radiation, and biotic factors and pollinators attraction. So these genes are, most of the genes were upregulated in the Woodwardia orientalis, and only few genes were downregulated in the for the flavonoid biosynthesis. So the conclusion is in transcriptome study, the functional genes related to vivipary mutant, spore abortion, seed development, seed storage proteins, calcium dependent, lipids metabolism, reactive oxygen species, plant hormone dehydration responses, flavonoid biosynthesis, and heat shock proteins were identified for the first time in pteridophyte research. In apomictic fern, we noticed the vivipary mutant genes NOT2, NOT3, NOT5 family isoform 2, and ABE content reducing gene, abscisic acid deficient 4 gene, and the spore dormancy reducing gene, transcription factor viviparous 1, and none of the genes responsible for reducing the spore dormancy, AB content gibberellic acid, and vivipary mutant genes found in the non-vivipary fern, Woodwardia japonica. So in the all these genes were present only in Woodwardia orientalis, that is a vivipary or apomictic plant, and none of these genes are present in the Woodwardia japonica. And higher expression of ABA, gibberellic acid inducing genes in non-vivipary fern, and lower expression of these same genes found in vivipary ferns. And the gibberellic acid is mainly useful for the germination of spores and seeds. So our study opened a new entry to the pteridophyte research for further research. Thank you so much.
Thank you, sir, for your informative talk. Students are requested to clear their queries. I request the students to clear their queries. No duty is more urgent than that of returning thanks. Now, I would like to welcome Dr. C. Anbarasu, sir, Assistant Professor, Department of Botany, Vikas, to propose a vote of thanks.
Good afternoon to all. It's my session. I would like to thank our most beloved chairman, sir, and his family for their fruitful support and blessings to conduct this program. Even though their absence, their blessings always with us. Thank you all. And I express my sincere thanks to our Executive Director, Dr. Kupusamy sir, for his guidance and valuable suggestions towards this program. I thank our Chief Executive, sir, Mr. Chokalingam, sir, for his support to conduct this program. I would like to thank our principal, sir, and our academic director, sir, for their valuable guidance, and fruitful support to conduct this program. It makes me to get immense pleasure now to thank our today's guest speaker, Dr. K. Bhaskaran Xavier sir. You made us to feel in the pathway where we can reach the goal which can be a role model for you to create a nice future, not only for us, to our future generation also. That kind of confidence really, sir, you made with your inspirational speech and fruitful presentation. The way you presented, really, it makes me to get, why don't you do the same thing in a different aspect? You said at the end of your conclusion part, this is the way where I initiated a new things. So in the same way, some kind of innovative things will happen in future also, sir. We are sure that. And I'm pretty sure the precious knowledge that you shared with us definitely help us to improve our students' future. Thank you very much for your enlightening speech, sir. Once again, I thank you, sir. And now I would like to thank our Dean of Bioscience, Dr. D. Shankar sir, for his valuable guidance to conduct this program. My sincere thanks to all the HODs of Bioscience for your support to conduct this program. And I would like to thank our department HOD sir, Dr. M. Kannan, who gave me this opportunity to give this vote of thanks. And I would like to express my deepest thanks to all the faculty members from various departments in bioscience. Without your support, nothing will be possible. So I would like to thank all of you. And not but not least, the achievers, the real successful persons, it's nothing but our students who are all going to make some innovation in future with the help of this kind of useful and valuable conferences. I would like to thank you all because you are the real pillars. Without you, nothing can be possible. And thank you all. Thank you so much for this opportunity.
Thank you, sir. Technical session 2 came to an end. After the break of 5 minutes, I request all of you to assemble in the same link. Thank you.
Students, stay here. Don't left from this. We have one more session.
Dear participants, we have another session. The session will be starts soon, so don't left in this group. Okay.
Students, please don't left. Please stay in. We have one more session. அனுஷா மேம் சார் வந்துட்டாங்களா?
இல்ல
அனுஷா மேம்
3 மணி வரைக்கும் தான் எங்களுக்கு கிளாஸே எடுக்கணும். ஆனா தேவையில்லாம எடுத்துட்டு வாங்க. சரி எடுக்கிறதாங்க சொல்ல முடியாது. பார்க்கலாம்.
Good afternoon. Anand Krishnan sir, are you there, sir?
மேம் சார் வந்துட்டாங்களா?
வந்துட்டாங்க சார். உள்ள ஜாயின் பண்ணிட்டாங்களா சார்?
என்ன நேம்ல இருக்காங்க?
ஆனந்த் கிருஷ்ணன்.
மேம் சார் என்ன ஜாயின் பண்ணலன்னு நினைக்கிறேன். ஐ திங்க் so. Please verify it.
ஓகே சார், நான் வெரிஃபை இப்பதான் கால் பண்ணி கேட்டேன். அவர் உள்ள இருக்காருன்னு சொன்னதுக்கு மறுபடியும் உள்ள வந்தேன். I'll check and come again, sir.
ஓகே மேம். ஓகே.
முன்னூத்தி 22 பேர் இருக்காங்க. நாத்தம் நீதானா? ஹலோ
ഇത് വരെ മീറ്റിംഗ് കഴിയടി.
Ansha ma'am, I didn't find the guest name in the participation list.
Hello.
Yeah, sir.
Hello. You hear me?
Yeah, sir.
Okay. You,
Yeah,
you
sir.
because you people are block my mute microphone.
Yes, sir.
That's why I couldn't.
When you are
Okay.
enter into the Zoom ID, you can give your name, sir, because we identified 2019 437
No, that is
660.
my university laptop. That is only going to pick up.
Okay, fine, sir. Now
Okay.
you can.
Anyway, no problem. You see now everything is okay now.
Yeah, sir.
Okay, I'm opening my video now. I mean in my camera.
Sir, we will have a formal
Okay.
welcome speech, sir, and guest. Are you ready?
Okay. You can see my slide there.
1 second, sir. Anand Krishnan, sir, please wait. We'll have a formal guest intro and welcome. Welcome.
Okay. Okay.
Ready?
Yes, ma'am.
Okay, to start.
A smile is the universal. Shall I proceed, ma'am?
Smile. You carry on. You carry on.
Okay. A smile is the universal welcome. With a smile, a spirit of giving, a sense of humanity, a pledge to spread peace and happiness, I welcome Mrs. M.D. Anusha ma'am, Head Department of Biotechnology, to give the guest introduction.
It's an immense pleasure for me to give the guest introduction in our international seminar. I welcome you, sir. I welcome Anand Krishnan, sir. He is from India. Now he is in South Africa being a member Royal Chemical Society, and he is a research scientist. He has a very big profile, which we have to take a lot of time, but I'll cut and short and sweet, I'll say. that he have finished his UG from Madurai Kamarajar University and PG from Bardiyar University, and he finished his PhD in Durban University, and he have finished his postdoctorate fellowship from Durban University of Technology, South Africa in November 2014 to November 2016. And now he's working as a medical research scientist in innovative postdoctorate research fellowship from the Department of Science and Innovation and also National Research Foundation, South Africa. He is now in Bloemfontein, University of Free State, Bloemfontein, South Africa, which is one of the leading university in South Africa. He have given a lot of publications and many scientific articles in international peer-reviewed journal, and also have has authorized many chapters as well as in review articles. He has awarded Best Postdoctorate Research Fellowship for 2016 and 2017 from Durban University of Technology. And he's an young scientist. He got Young Scientist Research Award in 2016 from Pearl Foundation. He is member of various editorial board of journal of international reputations. His research interest includes about organic chemistry, heterocyclic chemistry, medicinal biochemistry, drug discovery and delivery, extracellular vesicles, nanotoxicology, clinical biochemistry, and he has evaluated by the National Research Foundation and was awarded as Y1 rating, which is given to promote promising young. It's a pleasure to have you here, sir. And the section is open. We welcome the section.
Oh, done now.
Yes, sir, you can start now.
Okay. Okay.
You can start now, sir.
Okay. 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 Two things I want to ask you. First of all, my voice is clear?
Yes, sir, it's clear.
Yes, sir.
Okay, my slide is correct? My slide?
Visible.
Yes, sir, it also
Okay,
visible
okay.
clearly.
Okay. Good morning, everyone. First of all, I thanks to Dr. Arnagiri to invite me to talk his college, Vivekananda Women's College. Really, I'm really very happy to give my lecture to you people. Okay, today I'm going to talk about a small molecule with exosome controlling cell-to-cell communication. Okay, so this is my topic. Before that, I like to tell about my affiliation, where I'm working. Okay, my full name is Dr. Krishnan Anand. I'm a scientist in chemical pathology in the University of Free State, one of the leading university in South Africa. It also is aligned with the National Health Laboratory Service. It's a government clinical lab, which is linked with every university, helping to hospital. So my application with both. So I'm working with both institutes. Okay. Okay. Now I like to tell briefly about my background, about where I study in my master and PhD and postdoc. I studied in master in Bardiyar University under Professor Mohan. He is an eminent professor in organic chemistry. He's retired now. I was a master project student with his group. So I learned first my research methodology from his team. Then I got a PhD opportunity in Durban Institute of Technology in South Africa, Professor R.M. Gengan. So I did my PhD in Department of Chemistry in Professor R.M. Gengan. In 2014, I finished my PhD. Then I got a postdoc opportunity in the same group. Then 2 years I was postdoc with the Durban Institute of Technology. Then I moved to University of KwaZulu-Natal, one of the top university in South Africa, with Professor Anil Chotakan, who is a world-renowned scientist in toxicology. 3 years I was working with him. From this lab, I learned a lot of new techniques, which as a chemist, I became a clinical biochemist. From this research group, this is my turning point of my life. So these are my brief about my research and my university detail. Okay, you can see this slide. You can see the infrastructure of where I studied. Durban Institute of Technology is there in Durban city. Then Institute of Asal Natal is a Bill Gates funded institute. Then currently I'm working in the University of Free State with the medical doctors. They are medical students, not science students. They are MBBS students. Okay, so this slide you can see different type of peoples in South Africa. You can see the color is just for color, but this country is like that. This country is a rainbow country. It's a unity and a diversity. It's a very good example for unity. Okay, now here, how I started my research career. I started with the natural product based on plant. Then I started a synthesis. Then I started formulation. Then I started full into cancer biologist. Okay, so these are this slide is you can see this is a news from my where I did my PhD. So they put me in a newsletter because of in 2014 one of the best doctor student from that university. So they select I'm the best student from that university. So they put me in the newsletter. So these are my little bit. You can see some of the compound here. You make chemical structure here. So these are the early stage. I started research work with the small molecule synthesis and characterization and modification. Okay. So you can see the 2 books recently. I edited those 2 books, which is related to nanomedicine and diagnostic by using nanotechnology. Okay. Okay, here. This slide you can see organic chemistry, how I became nanochemist, then how I became a medical biochemist. Okay, see, I started with my career with organic chemistry. So the organic chemistry, one of the beauty things, the organic chemist can apply in any type of chemistry background research field. So you can see here natural product chemistry, biochemistry, catalytic chemistry, fluorine micro, they are linking multidisciplinary. So the organic chemistry can link with the different field because this is a basic mother of chemistry. Okay? Without organic chemistry, you cannot think about protein, you cannot think about molecules, you cannot think about the enzyme. So organic chemistry is very important role in the chemistry. So lucky I study in organic chemistry, so I easily understanding about clinical and the nanoscience, whatever in chemistry related, I easily understand because of the organic chemistry is like basic, like mathematics like that. So without organic chemistry, you cannot design any type of project. Okay, here you can see nitrogen heterocyclic chemistry. This is my master project. So I started a project with the nitrogen heterocyclic chemistry. So why is here nitrogens? That group, my Professor Mohan group, they most focus on nitrogen heterocyclic chemistry because of our DNA and RNA molecules the molecules similar to this, like you can see here, purine, adenine, guanine. These are base molecules. You can see that without nitrogen, this molecule not made of the other molecules. So the nitrogen is playing important role because of it's like acting as a base molecule. So we want if we want to develop the drug molecule, we want to interact with this molecule. So we developing same kind of molecule which is related to nitrogen based molecule. So we mostly focus on nitrogen heterocyclic. That group mostly focus on carbazole, and there's one of the derivative I did in the master indolquinone. It's indole plus quinine. It's called indolquinone. It's a very good DNA intercalator. you can see what is a DNA intercalator. You can the base molecules easily interact with the DNA. Okay, so it is disrupting the DNA. That means it's changing the DNA function. Then means it's stopping the cancer in a nucleus level. Okay, so we mostly designing this kind of molecule. So this was my master project. So I learned this from this lab how to synthesize molecule, how to design the molecule, how to characterize the molecule, how to purify the molecule. So I learned a lot of basic techniques from this lab. Okay, from Bharatiya University, Department of Chemistry. Okay, then I got PhD in Durban Institute of Technology in South Africa. Then I started the research career with the nanotechnology. So I learned a lot of organic chemistry. I learned a lot of techniques from organic chemistry. Then in myself, I designed project based on how to incorporate with the nanotechnology. I mean, organic chemistry to nanotechnology. So I designed the molecule which is can functional with the nanoparticle. So I choose one of the particle is a gold nanoparticle. So you can see we synthesizing molecule, sulfur-based molecule. Then I function with the gold surface for drug delivery or sensor purpose. I started my research PhD research project with the nanoparticle. Okay, you can see here synthesis of novel carboxyl and acid capped with golden nanoparticle. This is one of my chapter work which I published recently. 4 years before in Elsevier journal. You can see this is molecules. See this carbosol linking with the lipoxylic acid. See this carbosol knowledge I learned from Bharatiya University. Then the chemical modification I learned from them. Then what I did, I synthesized the same kind of molecule. The sulfur, I incorporate with the sulfur-based molecule. Then I'm coating with the gold surface. I'm using as a drug delivery is a drug this carbosol molecule. It's a very good DNA intercalator. So I started a project like that. Okay. Then after the gold nanoparticle, okay, I prepare gold nanoparticle based drug molecule. So then after that, after 2 years, I started to work with a different project, which is a self-assembly organic nanoparticle. Okay, so gold nanoparticle is an inorganic nanoparticle. Then I moved to organic nanoparticle, which is only contain organic molecules only. Okay, so you can see the aminocarbosol, lipoic acid, then I functional. Then this is called self-assembly is emulsification. Okay, so I'm dissolving with the water. High polar solvent. Then I'm mixing with the low polar solvent. So high polar solvent, low polar solvent is creating 2 layer. Then if you're giving us a proper emulsification reaction like stirring, it became nanoparticle. So small molecule, I'm converting into nanoparticle. Okay, so this molecule is atom level molecule. So I'm converting to nanoparticle, it's organic nanoparticle. So what is here you can see the TEM image of the nanoparticle. This is a mixture of this molecule is combined together, became a spherical particle. Okay, so this particle I'm taking to cancer cell line. We did quite good cancer studies. So I published this article. Okay, now, okay, these 2 slides. So this I'm just showing these 2 slides about my PhD work, nano formulation with the organic drugs molecules. Okay, now you can see small video clip how. So you can see that this is a nanoparticle. We binding with the drug molecules. This is like just animation how to understand. So we formulating the nano drug molecule, then we delivering to patient. So this is the receptor target method. Okay. So you see, it's delivering to blood vessels, targeting the cancer tumor. So it's exactly targeting the tumor cells. Okay, that's why we choosing the nano medicine. Okay. From this, we understand. If you take a pure drug molecule, maybe it will affect other normal cell lines. Or other normal tissues. So it will. That's why still now cancer therapy is still now is not successful, because the drugs molecules giving side effect. Okay, so we need to find out target which is we want to target. For example, lungs cancer. So we need to choose the lungs cancer cell line only, not the other like a kidney cell, other cells. Okay, so we need to choose our nano formulation which is to target to lungs cells. Okay, because every cell is a different function, different principle. So we need to design our molecule based on the lungs cell line, protein, or the biomarkers. So that's why this nano formulation we doing the research. Okay. Now exosome nanoparticle. Okay, now I'm entering to the exosome nanoparticle. Okay. Before that, I want to explain about. So I started research career with the synthesizing small molecule, the nanoparticle formation, biological application. Then I started research with the exosome nanoparticle, which is already our natural nanoparticle from our human body fluid. So this is also very interesting interesting and emerging research field, which is related to more into our human body function. Okay, so you can see this is a microscopy image of cells. You can see small particle which is it's called the exosome. These are cell membrane. This is a multi-vesicle body. So this is a natural. It's not like a cartoon picture. This is a natural cell image which is contained in the exosome. Okay. So why we is calling it a nanoparticle? This particle's size is below 100 nanometer. So we also is look like spherical, made up with the proteins and other biomolecule like DNA, RNA, mRNA, something, like a genetic material. So it's like a spherical model. So we call it a nanoparticles. Okay. Now what is the exosome? What is the principle behind the exosome? See here. Exosome is the range from 30 nanometer to 100 nanometer size, found in nearly all the body fluid like urine, blood, plasma, saliva. So you can find our body fluid, even breast milk, placenta. So you can find our all the body fluid. You can find this small particle. Okay, naturally it's forming. Okay, exported by urea cell types. Okay, exosome property reflect to the donor cell. Okay, see this point is very important role here. So every exosome have different kind of properties. For example, body fluid. For example, urine have different type of urine exosome have different type of properties which is related to kidney function. Okay, saliva have different type of exosome is related to our taste receptor, whatever related to saliva. Okay. So blood also have exosome. It is related to blood related immunogenetic related exosome. Okay. So even a central nervous system, our fluid have exosome. So that exosome related to the central nervous properties. Okay. So every exosome have depends upon our body fluid. They have different type of properties. Okay. Okay, these are the exosome properties. Okay, you can see the picture, the donor cells. Okay, the donor cells releasing the exosome, so it going to receptor cell. So then means you can see the exosome something carrying some message one cell to another cell. Okay, so if you see this picture, the exosome, the scanning picture, you can see here DNA is there, protein is there, surface protein is there, the genetic material is there. Then mean it carrier for genetic material one cell to another cell. Look like how father son taking genetic material from father, it taking to his generation, same thing like that. This exosome taking information, genetic material from this parent cell, it taking to other cell. Then means if this exosome carrying lot of message from one cell to another cells. Okay, so that's why this research, the exosome is emerging research to find out what is this inside. Okay, see, so far researcher developing drug molecule, developing like a diagnostic material without knowledge of this biomarkers, disease biomarker signaling. So now if we started study on the exosome, so we can find out exactly, for example, lungs cancer cell, how how delivering the, lungs cancer cells based exosome, what is the message taking to one cell to another cell. Like for example, this is a lungs cancer cell delivering the exosome, taking to normal cells. Then if we catch in the exosome, we study, scratch the exosome, we can find out the exact biomarkers for the lungs cancer cell. So if you found the exact biomarkers for a lungs cancer cells, lungs cancer cell, we can find the exact target protein and therapeutic target, okay, instead of hitting some other place. Okay, so that's why this field is very interesting. Okay, so you can see here a little bit when compared to previous, this picture is explain about little bit how that is exosome releasing and what is genetic material carrying, how they are interacting with the cell. So this picture is, you can see when the cell get apoptosis, the cell is dividing, so it is releasing small particle, that is the exosome is containing, you can see the DNA. So wherever the DNA is there, something genetic material is there. It's not only DNA, you can see mRNA is there, RNA is there, uncoding RNA is there, other transport membrane molecule is there. So these are genetic material carrying one cell to another cells. So if we characterize this genetic material, we can find out the exact disease biomarkers, and we can develop the new therapy, methodology based on this exosome. Okay. Okay, you can see this video how exosome communicating each cells. You can see here, this is a natural one. This is not the animation. This is a natural cells. You can see the one cell to another cell communicating to the nanotubes. That's, you can, if you see microscopy image, you can see some fiber. That is a nanotube. You can see the one cell to another cell, how the exosome communicating each other. So that means something is taking one message to one cell to another cells. Okay, so you can see. Okay. See, you can see the small particle going to this from here. See, it getting dividing. Okay. Okay. So now we can understand exosome is very playing important role in cell-to-cell communication. Okay. Yeah, here you can see this slide. This is one cell. You can see the red color spot is transferring to another cells. So something's carrying. Okay, so this exosome is a carrier. Okay, so if you isolate the exosome, if we characterize the exosome, we can find what is the exact biomarker for disease. Okay, not on any disease like Alzheimer or HIV or cancer, mainly the exosome playing role in the cancer. So so we can find out what message taking from one cell to another cell. Okay. Okay, application. You can see that exosome exosome is application is a different type of a multidisciplinary field. One is the biomarkers, another one drug delivery, another one targets. Okay. So yeah, as I discussed before, exosome carrying biomarkers. Okay, so you can, so if you isolate exosome, we can find out new biomarkers for new biomarkers. So based on biomarker, we can develop the drug molecule. We can design the drug molecule, so we can develop a new therapeutic method. Okay, so these are the target. Okay, so exosome containing proteins, RNA. So there we can control the exosome by using external like molecules, small molecule, which I am going to discuss later. Okay, so even we can use this exosome as a drug delivery carrier, like that, drug delivery agent like that. So we can load our drug molecule already FDA already in a natural drugs, whatever, like for example like a curcumin, whatever drug we have, we can load into exosome, we can deliver into cells. Because why we can this carrier, why is a specific, because we are isolating excess from the human body, human fluid, so it's more effective than synthetic carrier. Okay, because this carrier have some receptor, like easily the protein, it can find the directly in the cells when compared to other synthetic nano carrier. Okay, so this is very specific and target carrier. Okay. Okay, see exosome diagnostic. See, current diagnostics, you can in pathology, in research, researcher taking blood sample, urine sample, or saliva. Most of the diagnostic based on blood sample. Okay, so they're taking blood sample, they giving to pathology lab, they studying characterization of protein analyze or whatever is there. So they will say disease. Okay, this is a disease biomarkers we found from the blood sample or urine sample. Okay, so the patient have this kind of disease condition like that. So these are the current scenario diagnostic method. But future exosome diagnostic is going to play a very important role. Even you can predict the disease after 50 years, after 20 years, what that one patient, maybe you will get Alzheimer, maybe you will get cancer. We can predict early stage because the exosome is a genetic which is related to a disease also. Okay, so is one kind of, this is the research currently going on exosome diagnostic future direction. Okay, so exosome is a new method to diagnostic for future disease, like any type of disease. Okay, because the exosome mostly related to like nucleic material like mRNA, non-coding RNA. So we can predict before the disease coming. We can predict in early stage. Okay. Okay. This is personal precision medicine, which is what the precision medicine is nothing but it's not like if you take a patient, the patient have one disease, so we can design therapy based on the disease. The patient, the genetic and condition. Okay, so it's not like outside common for the medicine common for all. So that's not this medicine only for this patient. This is called precision medicine. So in future, we analyzing the patient history, like a small age to what what disease he have, what are the body metabolism he got. So based on the exosome, we can analyze the whole patient history. We can design the molecules. We can design the therapeutic for him. So we can give to the patient. Okay, so you can see the patient cell. We can isolating exosome from here. Then we can load into drug. Whatever. For example, if we have cancer, we can load into cancer drug. We can give back to him. So this one kind of future immunotherapy based on the patient history and the patient body metabolics. Okay. Now here you can see the exosome is a target delivery. As I discussed before, see exosome containing the some surface containing protein. The protein is a receptor protein. It is easily can bind with the cell surface. Okay, because the exosome is coming from cells only. So in this exosome, where can bind. Bind to select the cells, it can bind easily. So we can use this exosome as a drug delivery. Okay, we can load into our drug molecule. Like for example, I'm choosing this curcumin. It's a natural product. I'm loading into exosome. I'm delivering it to in cells. So this we can use this exosome as a drug delivery also. Okay. Okay. Now here. Exosome playing important role in cancer cell signaling. See, these are the factor of cell-to-cell communication. You can see neutralizing antibody checkpoint inhibitors and a direct connect. So exosome also one of the cell signaling important role in the cell signaling. So then means now Now we entering into our research field. So how to control cell-to-cell communication by using exosome. Okay. So for example, one vehicle taking a message to one stop to another stop. Intuitive in catching that vehicle, if you open that vehicle, if you finding what is there, so we can decide where we can decide what is carrying. So how to control that cells. Okay. So what I'm trying to say from this slide, the exosome is playing very important role in cell signaling. Okay. Okay, here. This slide you can see neticone sol is a long-acting antifunctional agent and orally active exosome secretion inhibitor. This is the one molecules already in the molecule only. This medicine only in Japan, not other country. So what they're doing by using this molecule, they inhibiting the exosome, which is related to antifungal disease. Okay. So this is stopping exosome secretion. See, that's why we want to inhibit the exosome. Okay. See, sometimes exosome do good things, also sometimes it will do bad things also. Okay. Exosome good things means we can find out new biomarkers and we can use for drug delivery. But sometimes sometimes exosome it will do bad things also, because for example, one patient have one disease. Okay. For cancer. Okay. When a cancer cell dividing, the cancers are releasing the exosome. Okay, what you will do this exosome? The exosome taking that cancer genetic material to normal cells. Okay, so it is affecting the normal cells. Okay, so this is a bad things for us. Okay, so if you inhibit that exosome by using some external things, by using small molecules, some protein, so we stopping the function of exosome. That means the normal cell is saving the normal cells. Okay, so only the cancer cells is not really. Finally, the cancer cells always to. Break. You need to release the genetic material to normal cells. Okay, so finally we inhibiting the exosome. That means we stopping the messenger. Then we stopping the cancer cell function. Okay, so by using the small molecule. So this is a very good example for already. This molecule is very good example for exosome inhibitor. Okay. Okay. Now, based on this kind of research method, I started my career as a chemist, as an organic chemist. I want to design the molecule to control the exosome, like by using the small molecule. Okay. So I'm studying the exosome properties. I'm studying the exosome surface protein. So I'm designing the molecule to control the protein. Okay, this is the recent project I've done so far. Okay. Okay, so this is the fusaric acid molecule I choose. Then I modified the molecule by using different. You can see phenolic hydrazine. The different derivative, one is a fluorine content derivative, another nitro content, one is normal like a benzene thing. So I designed the molecule, I synthesis the molecule, I characterize the molecule. Then we found the exosome. See, we do the another one research. We need to characterize the exosome. We need to find out what is the exosome surface protein, which is related to HSP19. Is a very important protein in the exosome surface. So human cancer exosome can see here that this protein more into cancer metastasis. So if you choose molecule to inhibit this protein, so that means we inhibit the exosome. So if you inhibit the exosome, the cell communication will stop in between the one cell to another cells, because the exosome is very important for cell communication. Okay, so we inhibiting the protein. So that means we inhibiting the cell communication. So we designing molecule and we binding with the this protein. Okay. Yeah, you can see the graphical picture of is to easily understand inhibit study of metastatic tumor derived exosome or non-exosomal protein. Okay. So you can see the cells. This is the cells is releasing for considered like a cancerous donor cell. Is releasing the exosome. This exosome may be carrying cancer genetic material to normal cells. Okay, so that is we isolating the exosome, we characterizing the surface protein. So there is one of the important. There are so many protein is there. There is a very important protein HSP90, heat shock protein 19. So we choosing this protein, we designing the molecule by using theoretical study like a bioinformatics study, like a molecular docking, and we doing simulation studies. So we theoretically we confirming this drug molecule can inhibit the protein. Then we going for a lab, cell line work. Okay. Before that, we studying the characterization, the structure of this molecule. Also, we doing intensity function theory, because we need to choose the molecule which is the easily electron donor or electron acceptor. So we need to study the DFT also. So this is the research method we doing. Okay. Yeah, here another one slide. This slide you can see same thing. The cancer donors are releasing the exosome, we incubating the cell. Then means the exosome in blood vessels that we incubating the blood exosome. That means it is inactive. The exosome is now inactive now, so it is stopping metastasis. Okay, because it's not going to transfer any genetic material to other normal cells. Also, not spreading the cancers to other organs. So that means by using the small molecule, we incubating the exosome to inactivate the function. Okay, so that means ultimately we stopping metastasis. Okay. Okay, now the another one surface protein is the exosome tetraphine CD81 protein. This is CD81 is one of the main exosome surface protein. See, so far I am working on exosome surface protein only. There are so many there inside is there. So many researchers doing different type of research. So, but I'm mostly working with the exosome surface protein, because as an organic chemist, I want to design the molecule to inhibit the surface protein, because I want to stop that the cells to cell communicate based on inhibiting the surface protein. Okay, so if you disrupting if you disturb the surface protein, then means you disturb the exosome, then means it will stop the cell to cell communication. Okay, so you can see the tetraphid membrane protein found in all exosome alongside other cellular ingredients and cellular contents. The tetraphid 28 protein is also known as CD81, belong to tetraphid family. CD81 are significant functions: cell growth, activation, development of some cell mediating signal transaction process. See, this protein very important on cell growth, activation, and mediate signal transaction. Okay, so if you target this protein, this three function going to disturb. Okay, so I'm designing the molecule to disturb this protein, exosome surface protein, to control this thing. Okay, cell growth, not a normal cell growth, is cancer cell growth. Activation, not is we disturbing cancer cells activation, and then we disturbing cancer signal by using that small inhibitor. Okay, this slide you can see recently I published this article with the designing the furanine molecule to inhibit the CD81 protein. So we did the molecular docking and the DFT calculation. We did the simulation studies. So this molecule showing very good activity with the surface protein CD81. So you can see this publication we published. Okay. So now, another one project what we doing now. So you can see cell derivative vehicle exosome bound monopartic interaction. Okay. So far we discussing the developing new molecule to incubate the exosome. This work is look like one kind of sensor, like how to develop. For example, we want to identify the exosome. Okay, to develop the like a label. We want to develop a new method detection of exosome, like like finding protein or DNA, like how we developing ELISA kits and protein assay. Same thing, we want to develop exosome kits. To find out exosome is there, the fluid like urine or blood sample have exosome. So we want to develop the kits. Okay. So by using the gold nanoparticle, I modify with the small molecules. We use colorimetry technique to detect that exosome. Okay. So here, okay. Before that, so gold nanoparticles. Everybody know about gold nanoparticles. Very good, surface properties and photonic properties. Okay. Because when we want to work with the sensor or a detector, based on colorimetric detector or colorimetric sensor, we need two things very important. One is the size and the color and the surface plasma resonance SPR. So this property is a very important role in the colorimetric principle. Okay. So I choose gold nanoparticles, one of the analyte, one of the detector. So I function with gold nanoparticle with which is easily binding with the exosome surface protein. Okay, so look like a signaling, like I'm taking the gold nanoparticle, I'm coating with a small molecule. That small molecule will bind with the exosome. Okay. Okay, you can see this the graphical abstract. How will we designing this work? I took gold chloride solution. I prepare a gold nanoparticle. Then I take methylene. This is a small molecule. You can see look like a amino hydrocarbon, like a glucose molecule like that. Then I coated with methylene gold nanoparticle, because before that, I'm not choosing just directly methylene. Before that, I did theoretical studies with the methylene. I'm binding with the CD63, HSP19. These are one of the exosome surface protein. So my molecule, I'm designing the molecule to bind the surface protein to inhibit the surface protein. That means the this golden nanoparticle color will change. So based on the color changing, we can detect that. Okay, we target exactly the CD63 protein or HSP19 protein. We catching the exosome. So that means our golden nanoparticle also catching. Okay. So here you can see the TEM image. You can see here. And the theoretical studies with the golden nanoparticle, how the binding with the gold protein surface. Okay. So this is just graphical abstract. I will explain again. Okay. Okay. So these are method to synthesis the golden nanoparticle. Okay. Synthesis of methylene golden nanoparticle, and I started binding of exosome with the golden nanoparticle with the different concentration and the characterization of the golden nanoparticle and application of the binding the exosome. Okay. So this is just a flow of our work. Okay, this slide is very important slide here. Improving binding efficiency. You can see this is a pure exosome. It's a TEM image. You can see if I take pure only the methylene, only without nanoparticle, if I'm binding, it's binding only two sides only. Okay, but if you coated with the golden nanoparticle, it's binding 5 place. So that means I'm improving the binding efficiency when compared to when without nanoparticle, with the nanoparticle, I'm improving the binding site. So you can see the TEM image. You can see the golden nanoparticle, the black color dot is the golden nanoparticle. These are exosome. So it is catching the exosome. Okay. So by using the nanoparticle, I'm capturing the exosome as well as we disturbing the protein. Okay, we inhibiting protein. So one kind of detector. Okay. See here. You can see this is pure golden nanoparticles, ruby red color. Then we are working with the low concentration to high concentration. So one of the concentration we saw in TEM image, the golden nanoparticle catching the exosome. Then you can see this high concentration. You can see little bit color changing. Also, you can see his aggregations forming. So you can see there are a lot of golden nanoparticles sitting here, but it catching all the exosome, is mixing the exosome now. Okay, so this is one kind of surface self-assembly aggregation principle. Is based on some physical principle typhoid introduced lean. Like if you see the pure golden nanoparticle, so if you take the exosome, it's changing the surface. Okay, so it's one kind of mechanism changing the exosome surface. Okay. Okay, now see, now this is by then. Okay. Okay, you can see this UV result. Okay, different concentration, that golden nanoparticle size going down. So we taking OD from there, then we brought in, we mathematically working on formulation, like find out rate, kinetics for the exosome. Different concentration, how is binding energy with the exosome protein. We finally finding a particular concentration, the gold nanoparticle can bind with the surface like that. Okay, then see here, I'm using a smartphone technique which is available in our mobile, by using color detector. Like see, these are, you can see the different color is here changing with the different concentration of exosome. The color also changing. Okay, so you this called you can get this app, like a paint color detector app available in our Google Store, Play Store. So you install that app, mobile app, so you can take the photo, so it will give different color with a different value. So based on our experimental, based on our UV experimental, we can compare with our result with this mobile application. This mobile application based on colorimetric algorithm. Okay, so I'm just making different result from. I don't want to follow other people normal, like doing same thing. So I'm just comparing with the recent technology. Okay. Okay, now so far I've done with my research work, which is small molecule, designing the small molecule and for incubating exosome protein. Also designing the nano formulation to detect that exosome. So far we discussed something about that, my research background. Now exosome visit, now current situation, so exosome research with the COVID-19 is coming up now. Okay, so now see the what is the relation between the exosome and the COVID-19. So exosome size is 500 nanometer, COVID-19 also like nearest almost 500 nanometer. Cell diameter is 100 nanometer, 100. The receptor also AC2 receptor and AC2 receptor. Okay, exosome also have AC2 receptor. Okay, is containing RNA, is also containing RNA. Is found in lungs fluid. It also found in lungs fluid. Okay, from this slide, we can hypothetically think exosome something related to COVID. Okay, so recently I submit one review article. Is almost going to accept. So if you isolate exosome from COVID patient or a COVID recovered patient, if you study the exosome properly, so we can find out novel biomarkers. Okay, so if you find out new novel biomarker from exosome which is isolated from COVID patient, so we can develop exact vaccine for the COVID. Not only exact vaccine, we can develop exact drug therapy also for COVID. Because of the hidden things we're not understanding about COVID function, COVID, the reality behind that COVID virus, which protein is affecting. So when only we thinking about spike protein, protease something. Okay. Anyway, we controlling that virus, but we need to design that how to kill that virus, how to stop that thing. We need to find out the exact biomechanism behind that virus. That means we need to find out the body, lungs fluid, which is generating from lungs. For example, COVID patient developing the exosome. If we catching that exosome, we characterize the exosome properly, we can find the new biomarkers for a COVID. Okay. Now you can see here, recently exosome companies coming up now. So all the pharmaceutical company now focus on exosome research because they're developing cosmetic, drug molecule, vaccine. So many things coming up, stem cell therapy, everything coming up now. So in future, exosome going to play important role in pharmaceutical company. Okay, so there's no more traditional therapy now. It's of advanced therapy, even hair transplant, and there's so many things coming up now. Okay, these are my reference. Okay, I'm done with my topic now. I just want to share about South Africa because this is 10 years I'm here. South Africa because I think I'm taking lecture with the young students. They want to know about South Africa for their higher studies. See, South Africa is one of the developed country. It's more equal to Europe. Okay, so people don't think of South Africa something far somewhere in continent, African continent like that. South Africa is a very developed country. Like it's more equal to Europe. So these are the city is there Cape Town, Port Elizabeth, and Durban. Currently, I'm working here Blue Fountain. I was in Durban. So it's a beautiful country. Also, is a modern country. Okay, English language country. It's not like other language. It's an English language country. Okay. Okay. Now you can see these are our people only came from our Tamil Nadu. So 150 years before they came from Tamil Nadu for a labor or to work in South Africa. So East Indian company they brought here for a contract base. So these are our Tamil girls. These are our Tamil young people developing the country by using their tribal track. The women are working the sugarcane field. So those time they very struggle. They're working hard. Now you can see this is also you can see this woman, the lady Valliyamma Mudaliyar. She is one of the actually I can say people do not understand or India people maybe don't know. She is the only starting Satyagraha initially before Gandhi. Gandhi followed her principle, then he started Satyagraha. Okay. So she started freedom fighting against two British people. She died in a prison. That time she was a 60 years old lady. She died in a jail. So then Gandhi came for a lawyer job. Then he taking our people unity. Then he started fighting against British people. So this is the building still is there in Durban. So he running news new Indian Congress something like that. So he running there. Okay, now see here. Now people how they are living now. See how I saw these people are now. Those time now people are their lifestyle changing because of the hard work. These are Durban city. Still they believing our Indian, our Tamil gods. Still the Mariamman temple. These are Mariamman temple. Still they keeping. Remember our farmers. So they driving mostly in a Mercedes Benz and the BMW. So still they following our culture. Still they marrying like our Indian, our Tamil culture. Yeah, this is one of slide. Okay, see you can see still you can see how we are in if you cannot believe here everything is here same culture. This is a Mariamman temple in Durban. Okay, still they taking Kavadi. So still Tamil and Hindi school also in South Africa. Okay. Okay, finally I'm coming to my thank you slide. See, okay, if you want to study in South Africa for PhD and postdoc, there are so many funding agencies there. Like that's called the NRF, National Research Foundation. This is the funding for PhD and postdoc. So you have to check every 3 months or 2 months one time this website. There, the call will come for collaboration, research project, and a PhD fellowship and a postdoc fellowship. So you can apply this through this funding. If you got funding, you can come to this funding. You can come to South Africa. You can start your research career in South Africa. So this is for our India. Indian student, TUAS and NRF doctor for undergraduate student. If they want to do PhD in South Africa, you can check this TUAS and NRF doctoral program. Monthly, I mean, every year one time the call open in I think June month. So you can apply. You can get almost 40,000 rupees, not 40,000, 50,000 rupees in our Indian money. So that is more sufficient to do PhD in South Africa. Also, NRF will open postdoc for PhD candidate. They open postdoc for postdoc research for our international student. They almost pay, if you got this funding, almost they pay almost 1,25,000 rupees in Indian money. So there's more sufficient to survive. Because South Africa is a modern country, as well as cost of living is very cheap when compared to Europe. Okay, so we can save a lot of economically we can stay here. Okay. So if anybody want to study or do research, if you want to do more information about South Africa, please contact me through email or my number, so I can help you for your higher education. If I know anything, I mean from other lab, any opportunity coming, I share with you people. Okay. Okay. Thank you. Also, I would like to thank Dr. Arunagiri, who is inviting me to talk here. Actually, I want to tell about Arunagiri. He was where I did postdoc. He was also postdoc. So we stayed in one street almost 2 years. So I learned a lot of exosome research things from Arunagiri also. Because he also working with the exosome. So I learned a lot of techniques from from him also. Okay. So thanks to everyone. I think I can finish now. Thank you. If you have any question, please ask me. Especially, I like to get question from students. Not only subject, any type of question for to do higher studies, or if you have any doubt related to research or other things, please ask me. I can explain.
Students are requested to clear their queries. I repeat, students are requested to clear their queries.
Students, you can ask doubts. Sir, thank you for your wonderful presentation, sir. I have a few doubts in what you have given. It was really interesting that paint color detector app that was very new to us. All the things what you have said is very new, and somewhat we know also it's very new. But paint color detector app, you said you are applying for this.
Yeah. It's a free app in mobile. Just for that is not only. I'm not using for only that one. I'm comparing my UV result to that mobile application. Just comparing, because nowadays smartphone sensor is a very important role in the future. Okay, so I'm just comparing my lab experimental result with my UV result. So if you see the paint color app, it will, if you catching the color, it will give one value, OD value. So that OD value I will comparing with my color, UV color.
Oh, you are taking spectrophotometer for OD values, or?
Yeah.
Okay, yeah, you are comparing that OD values with this paint color detectors, because
yes,
we are also willing to use this app for analyzing of this. Because
so you keep that. I mean, you can take that your color, your sample, take photo. It will give one value. That value you can compare with your UV value, so that it will match each other. So you can correlate both. You can plot the curve. Look, you can make it linearity with your UV result with your smartphone application. I think if you want, I can send some research article which is done with the UV spectrometry plus with the smartphone application, color up application.
Thank you, sir. One more, another question, sir. You said you have taken this exos with golden particle with various sizes.
What is
Yes, sir. How come you give it to this mice, or what is the organism you have taken, sir? Either it is in vivo or ex vivo, or you have taken any animal for that?
No, I didn't go for animal. Just isolating from cells. Sometimes I'm buying exosome because for detecting work, I'm buying standard exosome. I'm detecting, like look like a buying analyte, but I'm not taking from real sample from animals or something. I'm just buying from buying exosome. They have standard exosome, so I'm working with the standard exosome. I'm never worked with the animals.
Oh. You have not taken this animal cell line and extracted the exosomes like that. Have you done, or just commercially?
No, that one we doing for a protein incubation, not for detecting. For detecting, I bought the exosome.
Okay. In the end, you have said that about to COVID patients, if exos, you have said about that, right, sir? Where if we are going to do some research or something else in a wet lab with COVID, what is the organism we can take it? Either it is a guinea pig or mice, or because research is going worldwide, but we have some doubts regarding biotechnology like these things, because we are not. If you have any idea, you please share with us, sir.
Actually, see now nobody see there we for what type of research you want to do with the animals. For example, you want to initiate coverting with the animal, or just to the what type of research you want to do. Then only I can say.
No, sir. Actually, it's just a doubt I have asked you that whether for COVID. They are designing in silico with drug designing and vaccination preparation, all those things.
So far people are working with the hypothesis and designing the drug. You see so many paper coming theoretically, but still you need biosafety lab 3 if you want to do
in developing a vaccine or developing a drug in a preclinical and clinical sectors, usually we design a drug and we'll implement to the cell lines, and then we'll try it with humans, and then we move on to human beings. But due to this pandemic period, what happened is vaccines are erased, vaccines are given for human beings itself. But where is the time to give it in a research in an animal cell line, or they have tried it in an animal trial, or they have tried it directly to the human being, only in the cell line, and they moved on to the human being time being. For we all know that for developing a drug, it takes around 3 years minimum. But here in a very short period of 1 year, they have given they are vaccination. We also same field, vaccines are produced and it has been given. How come only they have tried in silico on cell line? Have they tried?
No. They tried with the animal. For example, commonly. The Johnson Johnson, COVID, this Covishield, that actually, so my knowledge, they're working with the SARS virus before, not the COVID-19. So they're working with the SARS virus. So the COVID-19 is a modification of the SARS. So they initially what they decided, okay, some of the vaccine, the pharma company already have a lot of formulation of the vaccine. So whatever they try with the SARS virus, they're taking the formulation. They try with the animals, which is like a monkey or something in the US. They tried with it. So suddenly they after so that's why last year, see, this is currently the pandemic. All the country decided to do no need regulation like FDA. We need to wait for regulation. So they started to immediately they want to do research with the animals. They started last year. Last year, I think this time we never think about the vaccine. We just thinking about maybe vaccine will come. So that time, so for my knowledge, I understand US, UK, they started with the animal. So then started with the, so when an animal giving normally other disease, it will take long process. FDA, the Food and Drug Administration, they're not processing the application. They're not processing properly. If for example, if I'm designing the one drug molecule for cancer, if I'm applying to get clinical trial from FDA, they will hold my application for almost 4 or 5 years. Then they will say, okay, go for monkey or go for other level. So they hold my application. But last year, what happened is a pandemic situation on the country. They accepted we need to do very speed process. So suddenly they test with the monkey. After monkey, suddenly they test with the human. So first they volunteer, they giving to volunteer for different different ratio. Like Africans and whites and ASEAN and South American. So they choosing some people, they tested, so it giving positive. Then they started. Okay, so still now I'm honest with everybody. Okay, I took 2 months before Johnson. Okay, I took Johnson. Okay, but when I'm Johnson, I took their farm one they giving. Okay, so the farm I am the one of the testing for them. But they tested already, but still they want to validate toxicity something. So I'm filling that form. Okay, I'm giving myself. Okay, you can test vaccine in my body. Okay, like that. But so initially, maybe they choose 50 people. Maybe the research center. Maybe they also giving sign with okay, you can test with me. So the 50 people success the vaccine. They go for 500 people. After 500 people, they go for 5,000. Now I am the 5,000 range, South Africa. So we got in Johnson from USA. They give, so that's why only the frontline workers we took vaccine, not all the people, not common people who's understanding about science and vaccine. So we are frontline. See, that's why you can see this is the National Health Laboratories. You can see my slide here. This is look like our India AIMS, All India Medical Research. It's like AIMS. This is South Africa AIMS, I can say like that. So whoever working in this lab, they are taking the vaccine, because we are working in a blood sample, urine sample, like that. So now so far last almost 2 months gone now. Now we giving our antibodies to test how we responding for antibody. Okay, so far everything good. No blood clot forming. Okay, since no single person forming blood clots so far, because Johnson very working good, because we are the testing people. So now government what they decided, now they going for above 60 years old people. Now next page they started. Okay, so same thing. I hope same thing. India also they applying the same thing, but I'm not sure. They testing with the SRM University. They testing with other research institute. Maybe they got a positive result. So then only they going for people. Okay. Hello.
Thank you, sir.
Okay.
The vibration of gratitude attracts more positive things into your life. Now, I invite Dr. M. Gauri Manohari ma'am to propose vote of thanks. Sir, I think that the particular staff is not available. Shall we wind up, sir?
No problem. No problem.
Okay, sir.
No, no need to. Thanks. Okay, so everything's fine now. So I can leave now. So I can leave now.
Yes, sir.
So can I leave now?
Yes, sir.
Okay, thank you. Thank you for all.
Thank you, sir.
Thank you.
Sir, system need. Yeah, participants, please leave from this meeting. Tomorrow we will join around 9:30 a.m. without 5.
அவங்க வந்து பேங்க்
Thank you all.
பிரின்சிபல் அக்கவுண்ட்ல போட்டாங்களா கான்ஃபரன்ஸ் அக்கவுண்ட்ல கான்ஃபரன்ஸ் அக்கவுண்ட்ல